Morning. Hello, everybody. It's Biology 105. This is October 6th. All right. We're in week 7. Thank you all for making it to class again. Yeah, recording's good. All right. Let's do a quick check-in because we're getting to about the halfway point in the semester, believe it or not. I mean, if you're not counting fall break or finals week, then it's a 15-week semester. You know what I mean? So we're basically halfway there. Is that working for anybody? Yeah, it helps me get through fall semester. I don't know. Great. So here we are in week 7. This week you're doing a classic lab. It's a polyphenol oxidase enzyme lab. You blend up some potato. Your TA blends up some potatoes, boils it, reacts it with a substrate. It's an enzyme catalyzed reaction. It's super classic. We're going to talk about enzymes this week in lecture, so it should sync up pretty perfectly. Next week, you've got the online, where is it? Lab midterm, okay? So we still have a lab next week to make everything sync up with Mark's labs as best as possible and to teach, the number of labs that we want to teach. I used to do an in-person lab midterm. I've moved it to an online option. This means that usually people do very well on the online lab midterm, all right? But don't forget that there is an in-person lab final, all right, that will be during our last week of lab. So, yeah, you can complete all of these with these questions with online resources, and you can answer the questions. I know this. You know this. You've taken online classes before. The first thing to check is just copying and pasting the question and then seeing if Google has an answer for it already. Fine. But that will not serve you on the lab final. So I really encourage you to try the midterm with your lab notebook, okay? And if you can't answer those questions with what's in your lab notebook, you need to think about how your lab notebook's organized, because I promise, every question can be answered with information in your protocols. If you're having trouble locating that information, those protocols need to get highlighted, you need to read them through a little bit, because in the lab final, you're gonna have 3 hours, but if your lab notebook's a mess, then it won't be the lack of resource that, tanks your grade. It'll just be who can maintain focus on a single lab final for 3 hours? Not many people. A question, please, and then I see yours there. I'll get you next.
Do you know around how many questions are going to be on the lab final?
On the lab final, I was just looking it over. I think it's like 20 questions, but there's not a very good metric because some of those are true or false. Some are multiple choice. Some are like, match the term with the temperature, And then some of them are, here's a population's genotypic frequency or phenotypic frequency, convert it into genotypic frequencies and tell me if it's in Hardy-Weinberg equilibrium, a concept we have not touched on yet, so that should sound intimidating. It is. It's a whole page, but it's one question. So I will tell you that in the past, with this lab final, with minimal changes, it takes some people 40 minutes and they get 100%. It takes some people 3 hours and they get 100%. Bless you. It really comes down to how much have you reviewed? Is your lab notebook organized? Do you have all the stuff you need? Solid? I love it. And you can always come talk to me in my office if you want to say, hey, can we look over my lab notebook? Can you tell me things that I should highlight in my lab notebook? I'll help you. I'll help you highlight. I'll highlight the dang thing for you, Just come talk to me in office hours. It always bumps grades up. Yeah.
Can you use digital notes on the lab final?
No. Everything must be physical. But if you've got a bunch of digital notes, you could print out an entire Wikipedia article, with tutorials. You can screenshot Khan Academy or YouTube tutorials and put those in your lab notebook. Anything printed, it's fair game, because in the real world, that's what you're allowed to do, is put whatever you want in your lab notebook. It's a reference guide for you, okay? I had plenty of printed out tutorials of how to do the equations that I consistently did when I was in my graduate school program. And they were really useful because I had to keep converting from ounces per hectare into grams per milliliter with a different application rate. And it was just like, oh, what it is. So, solid. Was there another hand up? If you are nervous about the lab final, well, hey, guess what, folks? I've got an additional resource that becomes available in week 5, and this is the lab practice final. This thing's 20 questions long. It's worth 20 points. Each question's worth like a point, but it covers everything that is on your lab final. So lab study guides, I have found, just don't work. Okay, I put a lab study guide out, people go, okay, Control F, I know I found it, I've got it in my protocols, it's there, I'm gonna do fine. So I'm gonna make you do an actual practice final the week before the lab final so that you are forced to confront any misunderstandings you have, all right? But it covers everything on the lab final. It's exhaustive, okay? I'm really, really trying to give you all the stuff I can for that lab final. Sick? It's my pleasure. Yeah, no big deal. No big deal. I just want to, let you know that that's why those things are there. Any questions now? I believe it or not, I am excited to start talking about enzyme kinetics. I just wanted to chat about where we are in the course. You're at the halfway point. You could have an F right now and still get a B in the class. Everything can turn around, It's only halfway through the semester. But check your grades. Check where you are. Be honest about it. Come talk to me in office hours. That's it. Enough of me. Enough of me. Here's some more of me talking about enzymes and energetics. Let's get back into it. So, good way to start, John. Good way to start. I believe this is the final slide that I threw up on the screen before we went to the weekend. We went into the weekend, all right? It's just representing flow of energy through the biosphere, bioenergetics, okay? The Earth is what we call an open system, meaning that it has an external energy input, and that's why we can get so much complexity on the planet. With no external energy input, everything kind of dwindles down to a stable state because every time energy changes forms, all right, some energy is lost. So the external source of energy for the planet is the sun, and it allows organisms and, I'll say organisms to keep it simple, to fight against entropy, all right? You got all these plants, they take solar energy, they generate potential energy in the form of these high energy molecules. You've got these organisms called the chemotrophs, we usually just call them the heterotrophs, other eating, all right? They consume those organic compounds, they break them down to generate smaller molecules, and they harvest the energy, okay? So what we've got is an example of an anabolic reaction and then a catabolic reaction, or a set of anabolic and a set of catabolic reactions in the biosphere, bioenergetics. All right, laws of thermodynamics. Cool? You cannot create energy. You cannot destroy energy, but energy can change forms. And every time energy changes forms, some energy is lost. All right? There's the bear. The bear ate some high-energy organic molecules. That's its fuel. It burns them through cellular respiration. It releases carbon dioxide and water. And then some of the energy lost is released as heat. Bless you. Cool. Entropy. What is it? Entropy is a measure of disorder. Okay? Every system falls towards disorder. If the system looks like it's trending towards being more ordered, you just haven't identified the energy input yet. All right? Every system, if you incorporate all of the energy transformations, is falling towards disorder. So while it looks like the Earth is generating order all the time with organisms growing and maturing and blah, 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 and all this stuff, it only looks like it's fighting against entropy because the sun, which is failing in its fight against entropy, because everything is, okay, is releasing enough radiation to drive those anabolic reactions, basically. So don't get too caught up in the gases having higher entropy than liquids, liquids having higher entropy than solids. That's something that can really start to screw with you if you really start to think about it. So I'd say don't think about it too much. We'll just focus on the definition of entropy, and we'll let your physics class deal with parsing this statement. Fair enough? Yes?
I always thought entropy was the measure of a lack of energy. Like the closer you get to like absolute zero, the more, the closer entropy or the more entropy has been attained.
That's enthalpy, which is part of the calculation for entropy. And really, just stick with me on this, okay? We really don't want to go down the entropy rabbit hole because it's really, 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 really fascinating and interesting, but it'll suck us in, and that'll be all we talk about for the 50 minutes.
There's like a whole other class that does that, right?
Exactly. Yep. So we're just going to talk about it very briefly, and we're going to focus more on like the energy transformations. Oh man, can you tell how hard I'm fighting against the temptation? Yeah. Let's keep moving. If I move forward a couple of slides, then I'll just forget about entropy. Okay? Energy changes. Okay. All right, cool. Whenever there's a chemical reaction, all right, there's some difference in energy between the reactants, the things that went into the reaction, and the products, the things that got produced by the reaction. Right? So you go from high energy molecules like glucose, and you break it down into low energy molecules like carbon dioxide and water. And that right there is, as we've already talked about, a catabolic reaction. It's also an exergonic reaction, meaning that it releases energy, okay? And when we're talking about energy being released, we're talking about free energy changes. It'll all tie together in a moment probably once we see some examples of this, okay? But we're going to be using this value, this delta G value, which is the change in free energy between reactants and products in a chemical reaction. Free energy is energy available to do work. It's another one of those definitions where it's like, just, if you don't mind, please just accept the definition because we need to talk about enzymes is what we're really trying to get into. And in order to discuss enzymes, we need to have a surface level understanding of Gibbs free energy. And yeah. So we'll just accept the surface level with me. All right, cool. So check this out, right? Here's like how you actually can calculate a change in energy, and it has to do with a change in enthalpy, the total energy, and the temperature, and is the temperature constant, and how much does the entropy change? So all these things go into calculating Gibbs free energy. We don't care, all right? We're in Bio 105. Take that out of here. Take it out of here, you know? Stick with super simple stuff. Exergonic, endergonic reactions. Some reactions release energy, they're exergonic. Some reactions require energy, they're endergonic. Cool? And Gibbs free energy is really just going to tell us, okay, whether the reactants or the products had more energy and how much energy the reaction required or how much energy the reaction released. So it's going to give us a quantifiable thing here, a quantity of energy required by the reaction or released by the reaction. Document camera. I should maybe put it on the big screen because the recording's capturing the big screen. Okie doke. exergonic, endergonic reactions, okay? And we're just gonna make some statements about them. So exergonic reactions release energy. Endergonic reactions require energy. Fair enough so far? All right, that's the definition of what these are. Exergonic reactions, well, that would include catabolic reactions. Endergonic reactions would include anabolic reactions. Okay, remember catabolics, or I'm just going to put this column as includes. Bless you. All right, so remember that the catabolic reactions break something down and release energy, whereas the endergonic reactions consume energy. I'm sorry, the anabolic reactions consume energy to build something up. All right, so see how these are all, interconnected. But then again, they might not always, every exergonic reaction may not be catabolic, okay? But every catabolic reaction is exergonic. It's very frustrating, I know. Then the delta G values, okay? So the delta G values are calculated as the energy of the product minus the energy of the reactant, which I'm going to show you in just a second on the slide, but I want to build a nice table with you that I can bounce back up, The delta G value in exergonic reactions, okay, is going to be a negative value because the energy of the product is lower than the energy of the reactant. It released energy, okay? So the product energy is lower than the reactant energy. So if this is 1 and this is 2, then our delta G is going to be a negative value. In the anabolic reactions, you guessed it, it's a positive value. Okay? And they tell us quantity of energy released. And this one tells us the quantity of energy required for the reaction to proceed. There's a nice big table. It's got all the stuff on it. Let me zoom in on it a little bit. Press the button. Press the button. Boom, 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 boom. There we go. Look at that. It's beautiful. I love seeing my disgusting handwriting, as tall as me. That's great. That's great. Legible enough for you there, folks? Awesome. So now what I'm going to do, let's see how this works, is I'm going to bounce this over to the side and take that back over here. And then we'll be able to look at the breakdown of each one of these points in the big table and provide further clarification. I don't know. That's how I thought it would go. So I'm hoping it goes. But will it go that way? Let's wait and find out. That's the exciting thing about every lecture. Things could go terribly wrong, Like maybe I got that wrong and then I'll have to correct myself, which is embarrassing. But that won't happen, right? Let's hope. Okay. Exergonic reactions. You're going to see a few of these sorts of figures. Nothing too wild here. The y-axis represents the amount of energy, okay? This is an exergonic reaction, and what we're saying is that the reactants had more energy than the products. Cool? Awesome. The difference in energy is the amount of energy released. Okay? So that's an exergonic reaction. Here's an endergonic reaction. Endergonic reaction requires energy input. The reactants had less energy than the products. You got to put energy in. There it is in the products. Cool? All right. So systems, okay? Or systems, we can describe them as having delta G values in their state, right? So let's not, I don't want to get, we're going to get a little caught up on this. Not too bad, though. This is just an opportunity for me to talk about open systems with an actual visual aid. All right? So check this out. The delta G value of this system is less than 0, meaning it is releasing energy. Okay? It's sitting at a delta G of -0 right here. Now, the energy of this system, the delta G value, is 0, meaning it's not going to release energy, it's not consuming any energy. The energy being released here is being used to light up a light. All right, very exciting. It's a little turbine, okay? So if you have a constant input of energy, you're looking at what we call an open system, okay, where there's an input of energy and the system can remain at a negative delta G, okay? You keep providing high energy reactants, essentially. All right? Here is a multi-step open system where you're providing high energy reactants and you carry out a breakdown and then a breakdown and then a breakdown. So you've taken the energy harvesting step here and you've broken it into multiple steps. That's really standard. We're going to talk about that in photosynthesis and in respiration, because usually, like, if you just take all the energy in glucose and you release it at once, then the cell boils and you perish. It's a terrible situation. If you've ever lit sugar on fire, it will burn, and that's just, you're releasing all the energy at once. It's no good. All right? Cool. So this will all come back very soon. So all this talk about reactions, as I told you, this table is entirely there so that we can, all these slides are here so that we can start to talk about enzymes. Because what are enzymes? Enzymes are proteins, and enzymes are biological catalysts, okay? So what an enzyme does is it speeds up a reaction, okay? Oh, yeah. Enzymes cause reactions to happen more. I'm trying to be very, very careful with my language because it's easy to use words like spontaneous or quickly, but neither of those are correct. I'm just going to use this. Enzymes encourage reactions, okay? And they encourage those reactions differently depending on if the reaction is exergonic or if the reaction is endergonic. So for an exergonic reaction, enzymes affect something called the activation energy, which I can sort of model using. As I can model everything, I will model this with my coffee thermos and my water bottle. Okay, here they are, 2 reactants. They're low energy. I put energy in to make a high energy product. This is an endergonic reaction. I put the energy in. Now there's energy contained here in my high energy product. And if I want to do an exergonic reaction to harvest that energy, it's not just spontaneously happening. Something has to input the energy to make it happen. So I have to put a little bit of energy in to make it happen, but then I can get all the energy out, all right? Or a good portion of that energy back out. So something has to activate the breakdown of the high energy reactant. And that energy input is called the activation energy. So what an enzyme can do is it could, for example, lower an activation energy barrier by destabilizing. I wish I had by destabilizing the reactant. Maybe by getting in there and just creating a slight strain on the bond holding these things together. So now it takes less energy to make this fall over. Does that sort of make sense? Okay, that worked. Awesome. Score. So that's how enzymes encourage exergonic reactions, because exergonic reactions are energetically favored. They don't usually, any energy you put into starting their breakdown can usually be harvested, and then some. An endergonic reaction, an enzyme works to catalyze an endergonic reaction by coupling an exergonic reaction to that endergonic reaction. And I'm going to explain what I mean there, all right, with our example of ATP. All right, here we go. What is ATP? Adenosine triphosphate. So it's got adenine, a ribose sugar, and a phosphate group. For a crazy amount of John points, let's say 5. What type of biological macromolecule monomer does this resemble? That's kind of true, but it's closer to another thing. Yeah, 5 points. Very good. Yeah, of course. There you go. So it's like a nucleotide, folks. Check it out. You know, A, T, C, and G are favorites, right? In the DNA code. Also, those of you who gave other answers, 2 points to each of you for being bold and getting out there. Very well done. Check it out. This and a sugar and 2 phosphate groups, and a phosphate group would be a nucleotide. Remember the sugar phosphate backbone from the polynucleotide in DNA? This is just one of those nitrogenous bases. So the biological system's being pretty efficient here, of like, hey, we can use, this with some extra phosphates as energy currency. And check it out, right? If there's an ATP, could there be a TTP and a CTP and a GTP? Probably. All right? GTP is very important. And these adenosine and these nucleotide triphosphates are very important in providing the energy for the synthesis of DNA. But all of that is an aside. I'm just excited to share that with you. And I'm stoked that we talked about it, but we're going to stop talking about it, and we're just going to talk about ATP. ATP is the short-term energy currency of the cell. The fact that it's similar to a nucleotide, who cares? Who's even worried about it? Not us. We got other things to be concerned with. Like, how in the heck an enzyme coupling an exer and an endergonic reaction has anything to do with looking at a diagram of ATP. Here's ATP, 3 phosphate groups, a lot of energy here. Right here in that terminal phosphate bond, ton of energy, all right? You snap that off, you're gonna release a really good, it's gonna be a really good exergonic reaction, all right? Check it out, here it is. Adenosine triphosphate, hydrolytic reaction, okay? Break off that phosphate group, you generate an inorganic phosphate, adenosine diphosphate, and release some energy. What the enzymes do is they couple this exergonic reaction and that energy release to an endergonic energy requiring reaction. And this is what provides the energy for most of those endergonic reactions in your metabolism, is ATP hydrolysis. Here's sort of a, like, here's a model of it, and I was going to say mathematically how it happens. The takeaway that your textbook wants from this figure is if the net delta G ends up being negative after you add the delta G of an exergonic and an endergonic together, if it's still negative, then the full reaction together is still energetically favored. All right? That's what they want you to sort of get from this. So let's look at the sections and hopefully it makes some sense. You're generating glutamine, all right? This is an anabolic reaction, taking glutamic acid and ammonia to smaller subunits and consuming energy to build up glutamine, all right? It has a positive delta G value, indicating it's an endergonic reaction, but you would have known that anyways because you're going from 2 little things to 1 big thing, and that takes energy. Fair enough? Awesome. Now, right here at the bottom, we've got ATP hydrolysis going on right here. Big to two littles, boom, boom, negative delta G, exergonic reaction. So check it out. Generating glutamine, it takes 3.4 kilocalories per every mole of glutamine. This reaction has a positive delta G value. Breaking down a mole of ATP via hydrolysis, that generates negative 7.3 kilocalories. Okay? Well, it doesn't generate that. It generates 7.3 kilocalories. The delta G is negative 7.3. Sorry, I got confused. Thank you. They were so happy. So check out, here's the math. There's the glutamine generation, positive delta G. Here's the ATP hydrolysis, negative delta G. And the net delta G is negative. So the free energy change for the full reaction is still negative, and therefore energetically favored. Okay? Now this center portion right here shows you, it shows you the phosphorylated intermediate, which is really frustrating to think about. But as a model, just for understanding, think about it like this. Okay, I'm ATP, and once again, these are everything we need to teach a bio class, which is just so shocking. This one has coffee in it, so it'll be my terminal phosphate group, because you know how much energy that contains. Ha 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 ha. We have so much fun here. All right, I'm ATP now. Hello. I'm holding out my terminal phosphate. There's so much power in that. There's so much power. Now an enzyme, okay, is going to couple a reaction, okay? What the enzyme can do is take that terminal phosphate group and temporarily, boom, attach it to this molecule. Now, this molecule, the table, contains more energy. It's at a high energy state, all right? It's what we call a phosphorylated intermediate, okay? Now, that phosphate group can then change the way that that molecule is shaped, or just provide the energy needed for an enzyme to snap it together with something else. So usually when you're coupling ATP hydrolysis to an endergonic reaction, there is some step there where the phosphate group gets transferred onto an intermediate. This is another one of those things. I've gone down the rabbit hole before on this, and I have found it not fruitful enough for a deeper discussion in Biology 105, but it is very interesting how the ATP hydrolysis coupled to endergonic reactions actually works. It's something that you'll cover in other courses if you decide to pursue it. Okay? A question, please.
Negative delta G is energetically favored, and this is because it's going to release energy?
Yes.
Okay.
It's because you're going from a higher energy reactants to lower energy products, and that's always favored if we're just accepting that systems always tend towards higher entropy, right? Lower energy states.
Therefore, a negative delta G is favorable.
Yeah.
Okay.
Yep. Negative delta G reactions are the ones that will be spontaneous, or that'll run away, like when something's burning on fire. It's just a negative delta G driving more negative delta Gs, right? It keeps providing the activation energy needed to break down more molecules of the substance. Of course, it's great, yeah. Okay. So if there's any takeaway from this figure here, it's really this, and the phosphorylated intermediate piece can sort of be neglected at this point. We'll talk about it again briefly when we talk about substrate level phosphorylation, which will be in our respiration discussion. Next slide. So, what can you do with the ATP energy? Well, you could do chemical work, all right? This diagram makes the phosphorylated intermediate thing make a little more sense, I think. Because look, you're trying to make the triangle go with the dang square. So you bind this P, and now there's a bunch of energy in that bond. And you push these things together, and maybe when that bond breaks, those will snap together. Oversimplification, but it's fine. All right. Transport work, pumping things. We've talked about that already. Mechanical work, make the motor protein make sense. Look at these. All of these involve you temporarily attach a phosphate group. And that phosphate group makes something change shape or just makes it go to a higher energy state where now the reaction will be net exergonic. Yes?
So ATP an enzyme in itself? Or is it
Great question. Is ATP an enzyme in itself? It's not. There's an ATP synthase enzyme that makes ATP, and then there's an ATP. There are a suite of enzymes that hydrolyze ATP and couple ATP hydrolysis to other energy requiring reactions. But ATP itself is not an enzyme because it's not a protein. It's got no amino acids in it. Every enzyme's a protein. Solid? I love it. Oh man, we're doing so great. Okay. Look, this is fascinating. Look, you put energy in, you get ATP. You break energy down, you release ATP. Solid? I love it. I love it. That was an emphatic thumbs up. And that sort of energy is just what we need. You know, just what we need. All right. Enzymes. So what we just talked about was how enzymes work to catalyze, to facilitate, to encourage endergonic energy requiring reactions. All right? Now we're talking about how enzymes work with exergonic reactions. Because as shown on the macro scale, when I stack the coffee and water bottle together, even though it's energetically favored for that to fall, it's not going to happen until something comes by and puts a little bit of activation energy in. All right? So enzymes lower activation energy barriers. In other words, they reduce the amount of energy required to start off the reaction. All right, so check it out. You've got the reactant and the product, going from a high energy reactant to a low energy product. The delta G value is represented by C. It's going to be negative, and it's going to tell us the amount of energy released by this reaction. Cool? There it is, boom to boom, awesome. But you have to put some energy in first. And that initial input of energy is going to raise the energy of the reactants because the energy can't just disappear, all right? So you put some energy in to the reactants and usually it's destabilizing them, okay? So you raise them to a higher energy state. The black line here indicates the amount of energy, so check this out. You put energy in, put energy in, and then eventually it breaks down, okay? Here, you put less energy in, and then it breaks down. This is the activation energy barrier right here, represented by A without an enzyme present, represented by B if an enzyme is present. It is key that enzymes do not actually affect the delta G value represented on this figure by C. Are there any questions about that? Yes, please.
So if the enzymes don't affect the delta G, like what happens to all that extra energy that's required to start it, like all of A?
Oh, yeah, totally. So the textbook conventional wisdom is you recover A. Think about and the analogy is there's a person on this hill on a bicycle, and they pump to pedal to the top of this hill, and then they coast down, and they recover all the energy they put in, all right? But really, energy is going to be changing forms here, so you're not going to get all of that energy back that you did put in. But if you have lowered the activation energy sufficiently, if you've made it low enough, then ambient energy levels, because any system, right, that isn't at 0 Kelvin has ambient energy, things are moving around. If you lower the activation energy levels enough, then just ambient present energy might be sufficient to kick off the reaction. So then it's like, what happened with that energy? Well, that energy was there and it's still there. You know, it just got re-radiated out of the thing when it broke, probably as heat. Does that kind of make sense? Awesome, I love it. But the real key here is that it's not going to affect how much energy gets released. No delta G change. Moving forward, how? How could this happen? How could this activation energy barrier be lowered? Well, the most straightforward example is putting physical strain, okay, on a bond. So you've got this thing that's gonna break down right here. Here's the reactant, it's gonna break down into products. There's the bond holding the subunits of the reactant together. The enzyme binds to the reactant and it just kind of flexes it, all right? It just destabilizes that bond a little bit. It could maybe put it into a chamber that has a different pH. It could maybe bond to different pieces of the large molecule and then strain the other bond. Or just by bonding, it can make that covalent bond, not exist anymore, right? And then once the thing breaks, the enzyme just lets it go. All right? So it was already energetically favored, so the enzyme just helps it break down further. Sometimes this involves an energy harvesting step, sometimes it doesn't. Sometimes the goal is just to break the thing down. Question?
Yes, enzymes get used up in the reaction.
They do not, and that is key, right? That these enzymes, they carry out the reaction, all right? They carry out the reaction, they release the product, and then they catalyze the reaction. They release the product, and then they can go back and repeat the whole process. So here's an example of an enzyme, okay? An actual model of an enzyme bonding or binding to it's the thing that it binds, which is called the substrate, okay? So we've got this enzyme, which is a big protein. The protein has a specific region called the active site. The active site binds something called a substrate. So the thing that it's breaking down is its substrate, okay? Really more accurately, the thing, and I'm sorry to be so vague with the language thing, but it's, it's fitting, okay? The thing that binds to the active site of the enzyme is the enzyme's substrate. So the cycle looks like this. There's an enzyme and a substrate, and they're floating around, and eventually the substrate hits the active site, and bing, now you've got this new complex formed called the enzyme substrate complex, indicated here. These square brackets, in this case, do not mean concentration, all right? It's like the only time they don't mean concentration. So this is the enzyme substrate complex. Now, the enzyme is somehow going to catalyze a breakdown of this substrate, all right, or a conversion of this substrate into product. Enzymes and the active site of an enzyme and its substrate fit together very, very specifically. So as soon as the substrate, represented here by my fist, okay, changes form, it no longer has an affinity for the active site, or the active site no longer has an affinity for it, more accurately. So then it gets released. The active site only takes circles. It helps to facilitate their conversion into rectangles, and then the rectangles are released. Okay? It's like this. This is all there is to it. It's just that simple, right? Okay. So enzymes and substrates come together to make an enzyme substrate complex. Then the product gets released, and then, to get to your initial question, the enzyme is free to start again and bind another substrate molecule. This is going to be really, really, really important because since these enzymes don't get consumed up and don't get gobbled up in each reaction, you have to regulate their activity tightly if you want to regulate your metabolism. So enzyme regulation is hugely important for biological systems to function appropriately, all right? And look at how much, look how big this enzyme is compared to its substrate. That took a lot of energy to make. You don't want to just destroy your enzymes in order to stop their activity. You want to regulate the enzyme's activity, a topic which we will talk about quite a bit. Cool. Oh, and here's the stuff I was talking about previously. about how the active site could, maybe it's like acidic, or, like by creating a favorable microenvironment, or maybe it just bonds to the substrate and takes the electron away, or something like this. Cool? Yeah, awesome. Hey, look, here's another example of this, where you've got the reactants going to the products, and there's like a step where everything gets destabilized. The reactants go into this transition state where all this energy has been put in, so it'll encourage them to, react. Activation energies. Okay, cool. Enzyme substrate complex. Just slow me down. These slides are about to get pretty repetitive, so I'm going to just kind of breeze through them, and you slow me down if you got questions. Okay, so I'm just going to get to the full thing. Check this out. Enzymes substrate is sucrose. The enzyme's called sucrase. Remember when we talked about that? How enzymes end in ASE and usually tell you the thing they break down. So sucrase binds its substrate sucrose. Boom, it fits. Boom, it strains that bond. Encourages hydrolysis. You get fructose, you get glucose, because now that they're not sucrose, the active site doesn't want them anymore. Released. Enzyme does it again. Cool? Cool. All right, great. So there's this reaction. This enzyme catalyzed reaction has a delta G of negative 20 kilocalories per mole. So the delta G is negative 20 kilocalories per mole. What type of reaction is it? Yep, it's releasing 20 kilocalories of energy per every mole of substrate that breaks down into product. So now I'm just going to double the amount of enzyme, and what's that going to do to the delta G value? Nothing. That's right, because delta G values affect activation energy. They do not affect, what did I say, delta G values? Enzymes affect activation energy. They don't affect delta G values, right? Okay, awesome. Yeah, the red curves with an enzyme, fine. Hey, look, it's like a volcanic hot spring, okay? This right here specifically is an acidic hot spring. These conditions are terrible for us. We would die. Our enzymes would not function here, okay? It's hot, it's acidic. I'm back, all right? Remember, we talked about protein denaturation. I've also told you that enzymes are proteins, okay? So extreme pH, extreme temperature, they cause proteins to lose their structure. And if an enzyme loses its structure, then it no longer has a functioning active site. It can't do its thing, right? Fair enough? So extreme environments like this one, okay, they do still foster life. There are those extremophilic bacteria and archaeans that survive here, but their enzymes are going to function differently. So different enzymes have different ideal temperature ranges and different ideal pH ranges, okay? So right here we've got, see look, this reaction didn't spontaneously happen, even though the delta G for that system was negative. Isn't that fascinating? All right, check it out. Two different enzymes, one from a human, one from a thermophilic bacteria, okay? For the human, the enzyme slowly increases its reaction rate and then eventually hits a key point where it denatures, and boom, the reaction rate falls down as the enzyme denatures. The enzyme can't do its job. Same thing here for this thermophilic bacterium, except that the point where the enzyme is most active is a higher temperature. It's all about its structure, okay? Now here's the thing, this is what I'm gonna finish up with. You're gonna see a very different curve when we look at enzymes and their ideal pHs as opposed to their temperatures. Here's why, okay? Remember temperature, all right? Remember heat. Remember how it's just a measurement of kinetic energy of molecules. I talked about that quite a bit. Check this out. Enzymes, okay, don't seek out their substrates. Rather, everything is randomly moving around, and every once in a while, a substrate happens to pop into its enzymes, to an enzyme's active site. All right? So as we increase the temperature of a system, you're increasing the likelihood that the enzyme catalyzed reaction can occur. So that's why you've got this ramping up right here, because as you get to higher temperatures, more likely that the enzyme will encounter its substrate. Here, it drops off because it denatures. So now it makes sense why the pH curve looks like this and the temperature curve looks like this. That's what we'll come back to on Wednesday. We will be done with our enzyme, the essential enzyme stuff that you need for Thursday by Wednesday. Keep up the great work. Keep coming to class. Keep being the best class ever. I appreciate all of you, and I'll see you again on Wednesday.